Welcome back. Can you hear me okay?
Yes.
Thank you. Thank you. Just checking, just in case. All right. So we're gonna move on to the second chapter of this unit. You probably noticed that this unit only has 2 chapters, but I know that these 2 chapters can be a lot of material for some students, or maybe seem scary to some students. Our previous chapter may have had a lot of vocabulary that was new to you, especially for those of you that had zero experience in microbiology before. And then this chapter that we are beginning today, even though in theory you should know some of the vocabulary already if you took cell and molecular biology as a prerequisite, or if you took anatomy and physiology as a prerequisite, maybe you didn't get it the first time. And this time, I want you to think of it as this is the last time you'll get to maybe try to get these concepts through. Now, obviously, the way we're going to go about microbial metabolism is going to be a focus toward the prokaryotic cell. Now, pretty much everything that we'll discuss is going to be at least told to you when it happens and where it happens in the eukaryotic cell. All cells, it doesn't matter what type of cell, have to have some sort of metabolism, some sort of system in place of enzymes in a certain order for certain chemical reactions to move forward. And that really is what metabolism is all about. Whatever that cell's DNA encodes for, whatever recipes that cell's DNA provides for, then those are going to be the only enzymes that that cell can make. And that is going to affect the types of metabolic processes that can take place. It may even be related to the type of environment in which these processes can take place. Now, this is all part of you to be able to understand energy and basically just how microbes use energy in the cells. And then we go on to the objective of metabolism that is going to talk about a little bit about anabolic pathways, and then everything is going to be catabolic, starting with glycolysis, then we go into respiration, and at the end into fermentation to get through all these objectives. Now, when we look at how we're going to go about it, we'll start with the basics of what is metabolism, the types of chemical reactions that take place. We'll talk a little bit about ATP. You should know what ATP is. You should have seen molecules of ATP before, so that's not new. And then we'll go on to enzymes, which is going to be, again, a review. If you took your prerequisites, at some point, someone should have mentioned enzymes. So we're just going to kind of go over them quickly and then really move on to metabolism. And we are going to go through the different stages, and then we'll talk about what happens with and without oxygen, depending on the types of cells that we're working with. Now, remember, we learned in our previous chapter that not all bacterial cells will be making energy in the presence of oxygen. Some of them will make energy without oxygen. And that is what I'm talking about when I say under certain environmental conditions, because some of these processes are anaerobic, Some of these processes can happen anaerobic and aerobically. And really, it's going to determine where and when these organisms can make energy. Now, if we look at metabolism as a whole, and this blue thing is meant to represent a cell, by the way, it's just a combination of chemical reactions. Well, anabolism includes the breakdown of the macromolecules for energy as far as it goes for us. Now, remember that our emphasis is toward the health sciences, and what you need to think about, Bowie, is that we have no photosynthetic microbes that can cause disease. So we don't usually talk about photosynthesis, and we don't even talk about that much about the processes that photosynthetic organisms that do not have chloroplast use at all, but we do mention the idea of photophosphorylation as a possibility, but it's not something that we go into detail. So if it's something about photosynthesis, which is just the breakdown, the conversion of sunlight into chemical energy, and the synthesis of small sugars, we don't usually cover it. But we talk about anabolism because without the breakdown of those molecules in catabolism, the cells are not going to be able to build parts. And they need to be able to constantly build parts because there's no molecule out there that is supposed to be eternal. So while the cells do catabolism, they have to do anabolism at the same time. It's supposed to be a balanced set of reactions. It's not true the way it works out in real life, but in theory, for each set of catabolic reactions, we should have a set of anabolic reactions, and it should be kind of like a cycle. It's okay, it's a valid question. Now remember, this very simple cartoon here, I tried to find the simplest cartoons I could find for these illustrations. It's meant to represent a cell. And cells are going to need, for most of the processes that take place in the cell, energy. And that energy is going to be in the form of a molecule that we call adenosine triphosphate, or ATP, which is what we have over here. Now, the way it works out, I always say follow the arrows. So in catabolic reactions or catabolism, we are going to see that nutrients that get brought in through the cell membrane into the cytosol get broken down. Now, first thing that you need to think about is this. The only molecules that will get broken down in those nutrients are going to be those that the cells have enzymes for. So try to connect this with what you have been learning in the lab. So in the lab, we talk about something like, for example, the citrate slants. And the only time a bacteria can cause a color change in that slant is because the bacteria had a certain enzyme called citrate permease that allowed for the breakdown of the citrate molecules. It was that simple. So if they didn't have the enzyme, then the citrate was untouched. So in catabolic reactions, that breakdown that is taking place is happening because that organism has cells that have the enzymes that can break down whatever that nutrient is. Now, the way it works out as a whole, and this is just an overview of metabolism, is that when these nutrients that are brought in get broken down, a lot of that energy from those nutrients, basically what was holding those molecules together, those chemical bonds hold energy, is going to be converted in ATP. ATP molecules. So we always try to think of ATP molecules as the currency for energy in the cells. Now, not all of the energy that was held by the chemical bonds in those nutrients gets converted into ATP because inevitably some of that energy is going to be lost to the environment and it is lost as heat. Now, those little building blocks that are needed for a lot of the anabolic reactions. Anabolic means to build something. Catabolic means to break down something. Come from catabolic processes. Many of the building blocks that will be used in anabolic reactions will come from catabolic reactions. And as you're putting small pieces and making larger pieces, you are building chemical bonds instead of breaking them down, then that is going to require that those cells use energy. And again, that energy is going to be in the form of ATP. Now, I don't want you to think that ATP is something like when you light a match and then it dies out and kind of like disappears and you cannot reuse it again. That's not the way this works out. Adenosine triphosphate, you probably already know this from your prereqs, is just a molecule that happens to have 3 phosphates. And whenever a molecule of adenosine triphosphate or ATP is used, all that's happening is that a phosphate group gets removed. So you're left over with the remaining molecule, which is adenosine diphosphate, and it will be through catabolic reactions that eventually, through many chemical reactions that will have the reattachment, if you want to think of it that way, of a phosphate group into the ATP molecules to rebuild ATP. Now, this process of recharging, maybe think of it as maybe a rechargeable battery, can happen multiple times. It has been measured in different ways. The numbers are somewhat consistent depending on what mechanism we use to measure it. But we know that at least a couple dozen times, the ATP can be converted into ADP, and ADP can be reconverted into ATP. And then what's going to have to happen is that, of course, more ADP molecules have to be built so that we can create more ATP, and that's going to be part of anabolism. Are there any questions so far? All right, now remember when we're talking about macromolecules here, it's going to be building the parts for the cell. Nothing in a cell is eternal, so it's going to have to be built or replaced at some point. That's where anabolism comes in. Now, as far as why the cells will go to the trouble of replacing parts, well, if you're thinking of a bacterial cell, you need to think of the functions of the different parts of the cell. The function of, let's say, the cell membrane for that bacterial cell. The function of the cell wall for that bacterial cell. Now, if you think of something like a cell membrane, you know it has a whole lot of phospholipids in there, and it has proteins, and it's meant to control what goes in and out of the cell. Now, if you were to be missing some of those phospholipids, or maybe some of those proteins that are meant to be part of the cell membrane of a prokaryotic cell, you can imagine that that function of control can be affected. Now, if we think of the function of the cell wall, which was what? What was that function of the cell wall in the bacterial world?
To control the entry and exit of things.
The cell wall. That was the membrane.
Oh, sorry. Yeah, structure.
Yeah, it's okay. It's okay. No, I get it because you were probably thinking about membrane. So structure protection. What do you mean by structure protection? What could the cell wall potentially protect against? That we learned about and we discussed in our previous chapter. Correct, osmotic pressure. So remember that having a cell wall seemed to be beneficial because when the water was going in too much, the cells didn't explode, those that had that cell wall. And when the water was going out, the cells lost a lot of the contents or the volume of the cytosol, but the cell stayed alive. They were just kind of waiting it out. So if you were to have, let's say, breakage in that cell wall, because it is made of peptidoglycan, and that peptidoglycan didn't get replaced, then you're going to have holes in the cell wall, and that function will certainly be impaired. So the cells are constantly building parts like that. Now, let's say though that the cells need to divide. They need to do that process that we learned about of binary fission. Because you remember that bacterial cells can only reproduce asexually. Well, they're going to have to double the entire contents of the cell. That is going to take a huge amount of energy. Are there any questions so far? Okay. Thank you. Now, because one set of chemical reactions, the catabolic ones, seems to feed the other set of chemical reactions, the anabolic ones, we say that in the cell, metabolism consists of chemical reactions that are coupled, and that the chemical reactions that lead to the production and to the breakdown of those molecules of energy, ATP, are coupled. So this idea of energy coupling is very important. And what I want you to think about is, if the cells use a lot of ATP, more than they're able to produce, because maybe they don't have enough nutrients going inside the cytosol, eventually the cells are going to start running low in energy and can potentially die because they may not be able to rebuild those essential parts. They may not be able to plug the holes in the cell membrane or in the cell wall that we were just talking about. Now, the types of chemical reactions that are involved that allow for this energy coupling to take place are going to be oxidation reactions, as you see here at the top, and then reduction reactions. And they're going to happen hopefully simultaneously. So we're going to spend some time before we go on to the entire process of, let's say, glycolysis, followed by cellular respiration, reviewing what reduction and oxidation reactions are. So I like to use very simple depictions that I can, the most simple ones I can find first, and then I can actually show to you where in the process something like this, where in the chemical reactions that we're going to go over, something like this can happen. So here's a very simple depiction of these paired reactions, oxidation and reduction. And again, it is very important that you follow those arrows. So this orange circle and this blue oval are meant to represent molecules. At this point, we don't care what the molecules are. We're just watching what happens whenever there is this combined or these coupled reactions taking place in the cell. So notice that we have the transfer of an electron from one molecule to another molecule, in this case the blue one, that happens to have space for that electron. Now that molecule that lost the electron or transferred the electron or donated the electron, I use all those words because depending on what you read, they use different wording, becomes what we refer to as oxidized. Now, the second molecule that took on that electron became reduced. And this is where sometimes some students have problems. They can't remember what an atom is, what makes up an atom, and some of the characteristics of atoms in general. So let's think of an atom of the element hydrogen, like what you have in H2O, which is water. An atom of hydrogen is going to have two parts to it. It's going to have a nucleus in the center of the atom, and surrounding that nucleus, there's going to be an electron cloud, or for now, just think of it as an electron orbital. Now, different parts of an atom have different characteristics. In the nucleus, you can find something that we refer to as protons, which have mass and have charge. And sometimes, besides the protons, you will find neutrons, which have mass but no charge. And then in this electron orbital or electron cloud that surrounds the nucleus, you just have these very, very tiny subatomic particles that we refer to as electrons, which have negligible mass. We don't count their mass. We only care about their charge that happens to be negative. The charge in the protons happens to be positive. These electrons are circling the nucleus only because they are attracted to that opposite charge that's in the protons that are present in the nucleus. In hydrogen, which is the smallest element in the periodic table of elements, we have just one proton in that nucleus and 0 neutrons and 1 electron. That's all there is. Just keep that in mind for now. So what I want you to think about next is, where is this electron that gets donated by this donor molecule coming from? It had to be part of an atom. It's not going to be just free floating. It's going to be part of an atom. At some point was part of an atom. That's what you need to think about. And it turns out that when we're talking about oxidation reduction reactions, like redox reactions, it's usually going to come from, guess what? A hydrogen atom. So at some point, that little electron gets moved to the acceptor molecule. And it's taking with it its characteristics. And its characteristics are going to be that charge. So what's happening to the molecule that donated that electron to that molecule that was the acceptor is that it's losing a tiny bit of negative charge. So it becomes less negative, which in chemistry is something that we refer to as oxidize. And that molecule that became the acceptor, in this case, the blue oval, took on that negative charge. It had space. And now it is less positive, it is more negative, so it is reduced. What you are reducing is just you're changing the charge. Something else that I always like to tell my students, you're also reducing the space for electrons that can potentially be held by that molecule. Now, are there any questions before we continue?
That very last sentence that you said about after it's more negative, it's considered reduced, and then what was the other thing?
So think of it as maybe having seats available, like if you're buying tickets for a concert, and that was the last seat available. There's no more spaces. It is reduced. All right. Any other questions? Now, if you're wondering, why are we spending so much time? The reduced one is taking something that's negative, so it cannot become more positive. It would be the opposite. So remember, we're looking at just the characteristic of what's getting shared, and the only thing that electrons have that we care about is their charge. Now, unfortunately, we don't get to do all the chemistry that you did in your previous classes here. This is just kind of like a refresher so that we can see why is it that we go through all this trouble. First thing that we need to think about is, why are we spending so much time talking about hydrogen atoms? Even though earlier we were talking about the breakdown of molecules, like, for example, glucose here, to make energy in the form of ATP. Like, how does this fit together? How do these hydrogen atoms fit together with something like, let's say, glucose? And I'm going to try a different approach this semester, which I know some of you are going to like, some of you may not be very excited about, which is making you think of what a molecule like glucose looks like. And I had thought about drawing the whole glucose molecule, and I know that some students don't quite like chemistry, but you should like it, because without chemistry, you do not have life. So if you look at the chemical formula for glucose, it is 6 carbons, 12 hydrogens, and 6 oxygens. These atoms that make up glucose are held together by chemical bonds. They're usually covalent bonds, which are pretty strong, which can potentially have a lot of potential energy that could be converted into ATP. So what I want you to think about then is we are going to break down glucose, but we're going to break it down starting by removing or by transferring some of those hydrogen atoms away from that glucose molecule. But we have a trick to do it. And the trick is not to just rip out the entire hydrogen atom. What we do is that we pull away electrons from the hydrogens one at a time. And what happens is really interesting. And here you have a more complex picture of what we were looking at. The protons that were left over that belong to that one hydrogen atom, so you have actually 2 hydrogens that we're going to be talking about here. What's going to happen is that, remember, the electron was attracted to the proton, which was positively charged. So it's going to be, they're going to be wanting to meet again. And simply what's going to happen in the cells when we're doing these sets of reactions of oxidation and reduction that we abbreviate as redox is that we are taking away electrons from the organic molecule, like for example, an organic molecule could be glucose. And at the same time, the protons that are part of that hydrogen are going to be chasing really after those electrons. And in the end, what you're going to end up with is one molecule that doesn't just have the extra electron, but it also has the proton that that electron came from. No. The chemical reaction involves the transfer of the electron. What happens after the transfer of the electron is that the hydrogens go away. So those are 2 separate events, that the protons go away. But you cannot have the entire hydrogen atom removal without taking that electron out first. Now, the reason why this is done this way, it is believed to be able to carry more potential energy to the parts of the cell where we're going to have the production of ATP. So the idea is this. If we were to go back to this picture that I've been using, if you were to rip out all of the covalent bonds that hold a glucose molecule together very quickly at once, without this methodic removal of electrons, most of that energy, if not all, is going to be lost as heat, because remember that even when we do this properly and we make ATP molecules, energy is lost as heat. So if we skip all of these chemical reactions, we are going to be losing a lot of that energy to the environment as heat. And this is not something that happens just in the prokaryotic cells. This is the exact same process that happens in eukaryotic cells as well. Now let's talk about this molecule here. The one that's actually picking up the electron and eventually ending up with extra protons. These are what we refer to commonly in the cell as electron carriers. In the cytosol of the cell, there's molecules whose job is to do this, to pick up electrons and subsequent protons that have been removed or transferred in redox reactions. Now, here's something that I also want you to think about. If you were to have a bunch of loose protons, H pluses, in the cytosol of the cell, something will change. What will that be? Make your AMP, make your AMP professor proud. Yeah, pH. So these molecules make it possible so that we don't have these weird shifts in protons in the cytosol either. So this is actually a very nice method of breaking down a molecule, so little bit by little bit, without necessarily affecting the environment of the inside of the cell. Now, there are different versions of electron carriers based on their molecular structure. One is the one that we call nicotinamide adenine dinucleotide, or NAD. The other one is nicotinamide adenine dinucleotide phosphate, NADP. It's almost identical to NAD, but it has a phosphate in it. And then there's the flavin adenine dinucleotide, or FAD. Now, so what you have in the parentheses in this slide is what they look like, or what we write them as, when they are in what we call the oxidized state, NAD plus, NADP plus, or FAD. That one is not a plus, by the way. After they become reduced because they have picked up electrons and their subsequent protons, NAD plus becomes NADH. That would be the reduced version of it. NADP plus becomes NADPH. That would be the reduced version. FAD becomes FADH2. So for what we're going to cover in this chapter, the ones that we're going to talk about, the ones that we're going to use are NAD and FAD, because these are going to be the electron carriers that are used by cells that are not doing any type of photosynthesis or any type of photosynthetic process. Cells that do photosynthesis have a third electron carrier, and that will be the NADPH. But again, we're not going to go over photosynthesis here. Are there any questions so far?
Dr. V, I have a question.
Okay.
So, for example, as NAD plus goes to NADH, it's only gaining one electron in that reduction, correct?
Good question. So let's look at that picture again. I usually don't say that until we start using them later so that it doesn't get too confusing. I like to think of it as baby steps, but you're asking, so I am answering. So this purple oval here is meant to represent NAD+, meaning before it picked up anything. It is still in its oxidized state.
Right.
Now, notice that there are actually two arrows going along. So what that means is that actually 2 electrons are getting transferred. And again, I'm just using this molecule name as an example. I'm saying that this is glucose. It could be something else. So what's going to happen, and this is what looks kind of funny here, but it's a lot more scary when I actually use organic molecule structures, is that one of those H pluses will become part completely of the molecular structure of that electron carrier. And that's why it becomes NADH, because it has now an extra hydrogen. But what I want you to notice is it picked up 2 electrons. Those red beads are meant to represent electrons. So there is this proton, this H plus, hovering really close to that NADH. And it will do so because it is chasing after the electron. And this is important because eventually we're going to get to this picture. I'm going to show it to you very briefly. That is something that we call the electron transport chain. And notice what's actually going to move and shift around here are bunches and bunches of these H pluses, of these protons. So those hovering protons, as well as the one that became part of that molecule, are all going to be traveling to the same area.
Sorry, my math is apparently pretty bad. So NADH plus has an extra proton, which gives it the positive charge, right?
That's what makes it become NADH. But
if you were to just add an electron, it would become NADH.
So okay. But remember, it is not becoming positive. It is becoming more negative.
Right.
Because it took the electrons. But it took 2 electrons.
That's where I'm confused. I don't understand how those charges align if it took 2 electrons. If it took 2 electrons and it was originally a positive, it should be now a negative charge, right?
Well, this positive gets basically taken care of by moving that entire proton over here. And having that extra electron, and they do have an extra electron, makes them more negative so that the protons can chase after them. So it is 2 whole H, 2 of these atoms that get transferred, but only one becomes part of the molecular structure of NAD+.
Okay, I'll think through that some more.
And this is why you have this NADH plus H plus, because this one did not become part of the molecular structure of that electron carrier.
Okay, thank you.
I have other pictures that have the entire molecular structure that I usually use in my cell and molecular biology class, which probably help you view this better. But usually I don't use them in microbiology because they tend to be a little bit more than what I usually talk about for this class as far as chemistry goes. But if you want to, I can send it to you.
That's all right.
It's a little messy, but I have it. It's a little messy, but I have it, yes.
That's okay. Thank you.
Okay. Any other questions?
So, Dr. V, what keeps that extra electron there if there's not a proton for it?
There was a proton in here. That's why this is positively charged. That was missing an electron.
Ah, okay, I got it. Thank you.
That's why it's a plus. That's why it's a plus, because it needs an electron. Anyone else? Now remember, the only reason we do it this way, that may seem almost really slow, it happens in nanoseconds, by the way, is because we're trying to keep as much potential energy in these electrons as possible so that we can then make those ATP molecules. We don't want to lose a lot of that energy as heat to the environment. Now, so you're probably wondering, why do we go through all this trouble of having these redox reactions? Remember, the end product, that what you really need to think about is, what is my goal through all of this, is to build energy. And that energy is going to be built in the form of these molecules of adenosine triphosphate. And again, if this picture here looks, because you probably saw it in cell and molecular biology or A&P, you have 3 phosphate groups. Here you have 1, 2, 3. And phosphate groups have a whole lot of oxygen atoms that are going to be electronegative and they're going to be repelling one another. But because there are covalent bonds between these, they just are formed, which is actually really good for us because that molecule over here especially has a lot of energy. And if you were to break off that 3rd phosphate group off of that molecule, you actually have energy that can be used by the cell to do some sort of work. And that's what our end goal is going to be. So let's talk about how we can actually do that. What is it that happens? How can we rebuild? How can we add phosphate groups to ADP molecules at some point through all these chemical reactions? And a lot of them are going to be these redox reactions that we just looked at, so that we can have some sort of stores of ATP somewhere in the cell. So there are 3 ways that we recognize that cells use to build ATP molecules. Now, anytime you add a phosphate to a molecule, it's something that we call phosphorylation. But for now, because we're thinking about it happening inside the cell, and we're adding an organic phosphate to a molecule that in this case is going to be a substrate, and that's going to be ADP. Let's just think of it as happening in the cell, not what you saw in chemistry if you took chemistry at some point already. So there are 3 different ways in which the cells can potentially attach that 3rd phosphate, as depicted here in this picture, to the ADP molecule. Now remember, I told you that a couple dozen times that can take place because there's recycling taking place. It's not a one-off type thing. This is not something that gets just poof, disappears in the cell. It gets recycled. So one way is something that we call substrate level phosphorylation. Another one is oxidative phosphorylation, which is what our focus is going to be mostly on for this class, these 2. For organisms that are able to do photosynthesis or some sort of photosynthetic process, then there is a 3rd possibility, and that is what we call photophosphorylation. Now, here's the deal. Some chemical processes, some metabolic reactions where you have several chemical reactions taking place one after the other in a specific order, are going to allow for substrate level phosphorylation to take place, while others are going to allow for oxidative phosphorylation to take place. Remember, we are ignoring that photophosphorylation for now. Now, in this picture here on the right-hand side, you have a very nice representation of substrate level phosphorylation. So with the help of an enzyme, which is going to be this purple blob over here, we're going to move a phosphate group from a substrate, which is going to be an organic molecule. In this case, it's PEP, something that is an abbreviation for a molecule called phosphoenol pyruvate. And we're transferring it to the ADP molecule. The enzyme is. The enzyme recognizes this substrate. The enzyme recognizes the ADP molecule. It grabs them, and then it catalyzes the enzymatic reaction, the chemical reaction of the transfer of the phosphate group from the PEP to the ADP. So after the transfer, this substrate, what used to be the substrate, becomes the product. And it has a different name because it lost atoms. It lost a phosphate group. It becomes pyruvate or pyruvic acid. But notice what happened to the ADP. It gained the third phosphate to become ATP. So with the help of an enzyme, we're just stealing away phosphates from one molecule to place onto ADP molecules. Where do we see this process? Usually, if we're talking about metabolic processes in the cell, in glycolysis, and again, we're going to see it in something that we call the Krebs cycle. So if any ATP is made in glycolysis, it's made via substrate level phosphorylation. The little bit of ATP that gets made during the Krebs cycle is also made via substrate level phosphorylation. And all that's happening is just the transfer of phosphate groups to ADP molecules with the help of enzymes. That's all it is. Now, it doesn't seem like a very effective thing if you think about it, because you're just having to find that molecule and you're just having to transfer the phosphate. All this work is only going to give you one ATP molecule at a time. But hey, it's better than no ATP. Now, the next type of phosphorylation that we're going to go over is oxidative phosphorylation. Now, this cartoon here is very simple, and it is how this takes place in the prokaryotic cell. Now, the way this is depicted here is very similar to what takes place in the mitochondria of the eukaryotic cell. So the first thing that you need to think about is this. Oxidative phosphorylation can take place in both types of cells. The next thing that you have to think about is in prokaryotes, it's happening in something that resembles a familiar cell structure. What does this look like to you based on what you learned in chapter 3? Try again.
The cell wall, membrane, blah.
Elizabeth, I think that you mean membrane and then you say wall, but I think you were right.
Know.
Correct. Yes.
I'm sorry.
No need to apologize. I had a feeling that was going to happen. So, membrane. Yes, the cell membrane, the phospholipid bilayer. So, just because bacterial cells do not have mitochondria, that does not mean that they cannot do the same things that eukaryotic cells can. They just do it differently or maybe in a different place. So they do this process of oxidative phosphorylation using the cell membrane because they don't have organelles. Now, just as a brief reminder, in case you didn't learn this, Cell membranes are supposed to control what goes in and out of the cell, so there's going to be possible movement from one side of the membrane to the other. And that is what's depicted here. You have all these black arrows showing you that there's transfer of molecules from one side of the membrane to the other. Now, you remember that membranes have a lot of phospholipids and they have proteins in them. And we had to have proteins in our membrane because not everything is going to be able to get through the membrane. Because if something is charged or if it's too big, it's going to need the help of a protein. And that's when we were talking about maybe facilitated diffusion, if it was from where there was more to where there was less, or maybe active transport. Either way, you're going to have one of those proteins to help you out. Any questions about that? I know that was just a review, but just in case. So, nothing is here labeled in a weird way. I mean, I don't want to say weird way, in a very specific way, because this depiction is just to give you a general idea of how this happens without it being too threatening. And that's why I like it so much. So that taking that into consideration, I want to tell you what the parts are supposed to be here, what are supposed to be the phospholipids, what are supposed to be the proteins, so that you understand. So these yellow and gray molecules are supposed to be the phospholipids. And then you have these pink squares embedded in between the phospholipids, which are going to be proteins. And then you have this large orange and yellow portion that is also going to be a very large protein. That's all it is, phospholipids and proteins. Now, let's talk about what's happening here so that we can have this attachment of phosphate to ADP to make ATP, which is phosphorylation. And we're going to talk about this process so that you understand why we call this oxidative phosphorylation and not something else. So here in green, you have a molecule. It could be, let's just say for now that it is NADH. It is NADH. NADH is what? Based on what we learned just today. What is NADH? An electron carrier. And because it has the H, what does that mean as far as what happened? What happened to the molecule? Yeah, it is the reduced version, meaning that it has the extra electrons and that it has extra protons. So here, these blue circles are supposed to represent the electrons that are being carried by that electron carrier. Keep an eye on those and notice what happens when the electron carrier NADH comes in contact with the first protein, which is that pink square in the cell membrane. Notice that it goes back to the way it used to be. It goes back to being NAD+. It's no longer carrying the electrons. There's no blue spheres around it. It transferred its electrons to that first protein. Now, what was it that was chasing the electrons previously, that was going on? What was it that followed that the electron pair to that NAD+ molecule that became then NADH? Besides the electrons, what was being brought over?
Protons?
Correct, protons. Now, the way it works out with the protons is that you had the protons that were part, if we go back to this picture, that were part of the molecule, here, that black circle. And you have the ones that are hovering around it. So what we're gonna look at is, where do they go in this picture? So this pink square is supposed to be a protein channel. And what we'll see is that the proton that was one of the protons, the one that was part of the molecular structure of NADH, is going to go through that channel, being that H plus is a charged molecule, to the other side of the cell membrane. And they're going to start accumulating at the other side of the membrane. Now notice that not all the protons went down that way. There are some over here. Those were the ones that were kind of hovering around that NADH. Remember, it was NADH plus one proton that was hovering around. I used to say, I mean, it's not very PC if you that the proton was so in love with that electron that it couldn't let it go and it had to chase after it. Anyway, so one of these protons is going to get to go through that first channel. The second one may go through the second channel. And what's going to happen is that we actually see a change in the concentration, a difference in the concentration of protons on one side of that cell membrane over time. So that you have more protons, more positive charges. That's where this oxidative comes from. We're producing oxidized molecules here on one side of that membrane compared to the other. The electrons, though, seem to be stuck to the membrane, and they are in a way. They get transferred from one protein to the next, to the next, to the next, to the next, until there are no more proteins to transfer into. So think of it this way. That first protein that takes on the electrons becomes reduced. When it donates its electrons to the protein number 2, it goes back to being oxidized. Protein number 2 became reduced. Well, it will donate its electrons, to the third protein so that it becomes reduced and it goes back to being oxidized. There's several redox reactions happening over and over and over and over and over, as long as there's a constant flow of these electron carriers that come in contact with that first protein in this chain of proteins, which we refer to as the electron transport chain. So remember, this NADH plus H plus, there was no room in that electron carrier to hang on to that proton, so it was just hovering around the negative charge. It's chasing after its electron. That's what it's doing. It's not part of that molecular structure, but it's very attracted to it because it is looking for the electron that it's lost. Now, it really doesn't matter where the protons are coming from here, whether they're the hovering ones or the ones that are part of the molecular structure of the electron carrier, because through these proteins that are used as part of the electron transport chain, some of them are going to allow for the movement of the protons from one side of the membrane to the other, so that you had different concentration on one side compared to the other. Now, remember here, there are no more proteins nearby to pass those electrons to. And what's going to happen is that a molecule on this side of the membrane will be there to pick up the electrons. If this process is happening in an environment that has oxygen, aerobic, oxygen can temporarily take on those electrons and it's going to become negatively charged temporarily. But what takes place is that, and this is what's really, really neat, when you have a difference in concentration on one side of the membrane compared to the other, what is the natural tendency for molecules? To always go from where there's more to where there's less. This very large protein that's a little away from the electron transport chain is going to be used as a channel that will allow for the diffusion of the protons from where there's more to where there's less to try to return the balance of the concentration of protons between the two sides of the membrane. But, and this is a really fantastic thing, as these protons move through the channel, they actually turn on the enzymatic activity of this channel. The name of this channel is ATPase or ATP synthase. And when that happens, when these protons are allowed to diffuse through the channel, every time a proton gets through, a phosphate is added to ADP to make ATP. And because what is driving the formation of ATP is the movement of these protons, we refer to this as oxidative phosphorylation. Now, some of these protons are going to encounter the now negatively charged oxygen. And what gets formed? Water, H2O, which is why one of the waste products of respiration is water. So every time you're making ATP, you're also inevitably making water molecules because of this. Questions so far?
So the electrons, after the electron transport chain, pretty much help to become water as a waste product. But the protons is actually what goes through that big channel to make the ATP. Is that what I'm understanding?
Yes, so the electrons are just kind of like a lure for the protons to get to this location.
Ah, got you. Okay.
It's a very nifty trick. But we need the protons. We really don't need the electrons. But without the electrons, we cannot get the protons to show up at this location.
Dr. V?
Yes.
Can you explain again what the those coenzymes are doing for those electron pairs to be transferred to the proteins in that chain?
I missed a word you said. You said explain again what was it the pink ones?
Yeah, what's going on with those with the electron pair that's to be accepted with the proteins in that bilayer?
So the electron pair gets donated from the electron carrier molecule, and this one, remember, was NADH, to that first protein. And that protein temporarily holds them. It becomes reduced, but very quickly we'll donate it to the nearby protein. And that protein is going to become more negative. That is going to make it more likely so that protons can get from one side to the other, because that positive charge and the negative charge inside the channel make it so that we can have that movement from one side of the membrane to the other. And the movement happens over and over and over again until there are no more proteins to donate those electrons to. And what's going to happen is that a molecule, if it is aerobic respiration, like oxygen, takes them on. Now, they're just there to facilitate these proteins that are here, the pink squares, are there to just facilitate the movement of the electrons so that none of these proteins stays stuck in that reduced state. We want to continuously have flow of electrons here so that more and more of these electron carriers can come in and drop off electrons. Because only when that takes place, we're going to receive the protons that are going to get sent to this side of the membrane so that they can diffuse back through that channel.
Okay, so this is like a whole system that doesn't stop. So this is always going on, right?
Yes. Okay. So that's a good point. So is this supposed to ever stop? The answer is, if it was to stop, the cells will immediately die. Like, it would take nanoseconds for them to die, because if you stop the movement, let's say here, even at the end, if there were no oxygen, actually, let's think of it as what happens if you were to put bacterial cells that are exclusively aerobic, that can only make energy in the presence of oxygen. Let's say that you put them in an environment that has no oxygen. This electron receiving protein here is not going to be able to donate its electrons to anyone because there's no oxygen around. And the process will completely stop. And you can see that what's going to happen is that no ATP will be made at all. And the cells run through a lot of ATP very quickly. They're counting on this process to be continuous. So if there's anything that stops the flow, then no more ATP can be made. And by the way, there are some cellular poisons like cyanide that bind to proteins in the electron transport chain and that's how they kill you. That's how cyanide kills you, by the way, when you intake it, because your electron transport chain receives the cyanide molecules. And that last transport protein stays permanently reduced, so this flow cannot continue. So the question is, do I know how many water molecules are made from respiration alone? So the easy answer that I can give you without giving you specific numbers, because it's going to be this. In theory, for every ATP molecule that is produced, one water molecule should be produced. But it's more, by the way. But it's more. That would be the minimum. It really depends on what cells we're talking about and the environment in which that cell is living at. Now, notice that I'm using this aerobic word over and over again, and that is why I like to cover that chapter before we go into this chapter, because you know that that is not the only possible type of oxygen requirement that we see for bacterial cells, for microbes. Sometimes you have microbes that are strict anaerobes. And if they are indeed strict anaerobes, then whatever is accepting electrons, what we refer to as the final electron acceptor. In that case, it will not be oxygen. It will be something else. And whatever that is varies depending on what microbe we're looking at. Questions?
So the electron carrier, once it drops off the electrons to the, it just goes back and picks up more electrons, or does it go through a different process?
So electron carriers are just going, they're kind of like Ubers or Lyft drivers of electrons, if you wanted to think of it that way. They're dropping off electrons, picking up electrons, dropping off electrons, picking up electrons, and they can do it 2 to 3 dozen times.
Okay. And then the only other question was, so if there is no oxygen, then that's another process.
So that would be anaerobic respiration. It's just that instead of using, that would be instead of using oxygen, you use something else. If possible.
Got it.
Now, remember, there is an N, and this is where I tell you that by explaining oxidative phosphorylation, you have already learned, without realizing it, electron transport chain. Because that is the process, oxidative phosphorylation, that exclusively takes place in the electron transport chain, which is in the cell membrane of bacteria and in the mitochondria of eukaryotes. Same system, it works the same way, it's just location is different. Now, by the way, this process is always going to yield more ATP than the other type of phosphorylation, because here you're doing it one at a time. Now, you need to think about it this way. For each time a proton gets pushed from one side of the membrane to the other, that will be a possible potential proton that can get passed through the ATP synthase to make ATP. But we're going to talk more about that when we go on to the details of the electron transport chain. Now, remember that there was that third electron carrier that we talked about earlier that I mentioned, and that there is such a thing as photophosphorylation. That is when a photosynthetic organism can potentially make energy, make what we just talked about, that process of osmosis, of diffusion of protons, which we call chemiosmosis, take place with the help of sunlight. But we're not going to go into the details of this. Now, if you took cell and molecular biology, you learned about photosynthesis, and you learned about how all of this works. All right. Now, what we're going to do next is talk about the different stages of metabolism, starting with digestion. So before all of that can happen, digestion has to take place. We have to break down the molecules. After digestion, then we can have something that we call glycolysis, and then we go from there. So remember, this is something that I mentioned earlier today. The cells, the bacteria, like those that you see in this plate over here, are only going to be able to use something as a carbon source if they have the right enzymes to do so. And why is it that they digest something? Why is it that they want to break it down? What are they supposed to eventually make?
Energy.
Energy. What could they use those monomers, those parts that are left over for? If we go back to that first picture, what could they use the parts of what they break down for? Whatever remains.
Storage.
Yeah, but before you store something, what has to happen? You have to have synthesis. You have to have anabolism. So whatever is left over, if it's not used to make energy, it can be used to build parts for the cell. And we're gonna pause here for today before we take our break for lab. Are there any questions before we stop for lecture? When I looked at it in the calendar, it looked okay. So all of the due dates are going to be the due dates that you have from your last syllabus. Now, our homework number 3, if you look at our last syllabus, it's not due until after. So if you're looking at homework number 3, which is the one that has to do with our next unit, not this unit, the next one, that one is not due the same way as it was before, because if you look at the new version of the syllabus, it is due after spring break. So I need to check on that. Something is funny because when I looked at the calendar, it looked okay. So something got broken. It's possible. I'll look at it. Anything else before we pause? All right, I'll see you in a bit.